Hello? Maybe I should remove that first. Okay, should I begin? I guess so. Good afternoon, everyone. My name is Ioannis Michalopoulos, and today I will be presenting HGCA2, which is the short name of Human Gene Coexpression Analysis 2, which is a website, an RNA-seq-based website for coexpression analysis in human genes. This project was carried out in the Biomedical Research Foundation of the Academy of Athens in the lab of Dr. Michalopoulos under the co-supervision of Professor Ioannis. So first, we should mention what gene co-expression analysis is about. So in gene co-expression analysis, we combine unrelated transcriptomic samples, which are from the same organism, in our case Homo sapiens, an experimental platform, but those samples actually can be from different tissues, developmental stages, and experimental conditions. Then we get a list of expressed genes, which are actually genes which have the same expression pattern in a multitude of samples. And we see that co-expressed genes actually tend to participate in similar biological processes or metabolic pathways. So this is the main HGCA2 workflow, and we'll be going through it step by step, and I will be explaining on the way. The first step, of course, was our data acquisition. In this case, we selected the GTEx public repository of RNA-seq data, and the good thing about GTEx is that all the samples are prepared by them using the same experimental conditions and experimental protocols. In addition, those samples are meticulously annotated, so we know everything about them. We decided to download the already processed gene read-count data, which corresponds to around 55,000 genes from 17,000 samples of 52 tissues. There were initially 54 tissues, but we decided to remove the cell lines from about 1,000 healthy donors. The next step is the normalization of those samples, so we can have similar values in each sample. Although there are many different ways to normalize RNA-seq data, we decided to use the most novel Q-smooth algorithm, which is short for smooth quartile algorithm. This algorithm, the interesting thing about it is that it makes two certain assumptions. The first one is that the statistical distribution of each sample should be the same of each sample within certain biological conditions or groups, in our case tissues, should be the same. And the second assumption, which is an assumption made by most normalization algorithms, is that the samples, the statistical distribution of all samples should be similar. So Q-smooth then takes the average out of these two assumptions and produces our gene expression values. We then store those values in a MySQL database, so it can be easier for us to access and use later on. The next step is the filtering of our samples and getting a representative list of samples. As you can see right here, it's not really visible, but anyway, I will explain. We get an expression value that corresponds to a certain gene and a certain sample. So, by using the expression values that we stored earlier, we are able to calculate the otherwise Pearson correlation coefficients for our set of samples. Then those pairwise coefficients are distance transformed, and we get a finely formatted distance matrix of samples. This matrix is then used as input to UPGMA hierarchical clustering algorithm and this produces us a correlation tree of samples. Then, by using our own method of auto-pruning, we trimmed the samples to about 20%. Now let's zoom in on that for a minute. So this is the actual tree of the 17,000 samples. You can see how all of them are grouped, and the interesting thing is that samples of the same tissue are actually clustered together. In addition, we can also see the results of our pruning, which is demonstrated by the long lines. So actually, the long lines of the tree are the samples that were retained after the pruning, so the 3,500 representative samples. Moving on, the next step, of course, is the actual Coexpresssion analysis. By using our 3,500 representative samples, this time we produced the pairwise correlation values between each of our available genes, which is once again distance transformed. This time a gene distance matrix is produced, which is then used as input, likewise to give it a hierarchical clustering algorithm. And this produces our final gene expression tree, and in this tree, each leaf actually corresponds to a single gene. So we expect genes who are co-expressed with one another to be in neighboring clades. Moving on, in order to discover the predominant biological processes that correspond to a certain list of co-expressed genes, we performed biological term enrichment analysis. This is done by utilizing the hypergeometric distribution, and then the resulting p-values are FTR adjusted. We offer a lot of different enrichment categories, from gene ontologies to gene disease associations to biological pathways, protein families, and last but not least, we are also unique in offering gene targeting transcription factors, which are also experimentally verified. So this is the main HGCA2.0 website, and you can see that it's available both in Elixir Biotool's website as well as our own home website of Michalopoulos lab. So here the user starts by typing a gene of interest, and by selecting the certain gene of interest, the webtool produces part of the total co-expression tree. We call that a co-expression subtree, which contains the gene of interest color-coded in yellow, and its co-expressed genes are in the neighboring clades of the subtree. The user can also perform all kinds of enrichment analysis right here. So then I will be presenting some indicative examples of HGCA2. So in the first three trees, my cursor is not really visible, but anyway. In the first three trees on the top left, the driver genes are co-expressed with genes of the same family. You can see the metallothioamines of the first three, the complexes of late conified envelopes, which are skin development, and the other two trees on the bottom and on the right we use driver genes which are co-expressed the resulting trees by using the certain driver genes are co-expressed with a functional partners. So in the bottom tree, we use a histone protein which is co-expressed with other genes related to response to stress and stress from heat. And on the second tree, on the right, we used NFKB2, and you can see RelB, which is its partner forming a heterodimer, as well as BCL3, which is ecoactivator of the NFKB Homo heterodimer. Sorry. Next, I would like to present a more interesting example. In this case, we use STAT1 transcription factor as a driver gene to HGCA2, and it produced a Coexpresssion substrate which contained many interferon-induced genes. Then, on the resulting enrichment analysis of this substrate, we found out, the HCA rather found out that most of the terms are related to the immune response to virus, including hepatitis C, influenza, and most interestingly, coronaviruses. To add on that, we can see on the red square on the tree that the OAS family of genes is also expressed with STAT1. OAS, especially OAS1, were found to be overly expressed during COVID-19 infection. Furthermore, certain mutations in the OAS1 gene, certain SNPs, were found to be correlated with severe cases of COVID-19 symptoms. Last but not least, I would like to show down there that the STAT2 transcription factor also seems to target the co-expressed genes. And this is really important, as it seems that STAT2 is the master regulator that drives the transcription that is shown right here. The transcription, the co-expression that is shown right here. We also used that list of co-expressed genes as a way to perform comparison of HGCA2 with other similar co-expression webtools that are also RNA-seq based. So, in each case, we use STAT1 as input to each of the other three websites, and we got the top 34 co-expressed genes with STAT1. Then we used each list as input to string to get 4 different protein-protein interaction networks. And judging by the network density, it seems that HGCA2 outperforms the rest of the tools. So, to conclude, I would like to say that HGCA2 can discover potential functional partners to gene of interest and assign biological roles to gene of unknown function through a guild by association approach. Since known already established bibliography has shown that HGCA2 can indeed reproduce known biology through its results, that means that HGCA2 can also produce working hypotheses for which are then to be experimentally verified. That makes the tool especially interesting to molecular biologists and experimentalist biologists. And this is the reason that we have made the GUI, the graphic user interaction, really simple and easy to use. And we also try to give as easy and understandable results as possible. Finally, we have also developed an API in order to make our results easier to be extracted. I would like to thank my supervisors, Professor Economidou and Dr. Ioannis Michalopoulos, as well as the rest of the author team that have developed this website, including Tolis, Konstantinos, and Christos, and of course, the rest of my colleagues in the Dr. Michalopoulos lab, especially Ellie and Joanna, who helped me with the presentation. Thank you very much. Okay, first let me clarify that in this case, we wanted to do a condition-independent co-expression network. This is exactly the reason that we used GTEx data, because you might know that already, that the samples from GTEx derive from healthy donors. So even if we wanted to, it wouldn't be really easy to perform a condition-dependent or rather disease co-expression network from those data. Now, could you repeat the second part of the question? I mean, you said about why...
Yeah, in any case, even if you use only health samples, it could be quite heterogeneous, the input sampling. So, okay, it's easier to understand in the case of disease and health issues. If it is meaningful to split first the data sets in different categories and then find the correlation for expression there, and then decide using a different algorithm whether two genes should be linked together or not.
I mean, we kind of did that in the case, this is why we used a representative amount of samples, because we wanted to remove any tissue bias, if you understand me. We didn't really want to have any representation, any tissue representation. We wanted our data to show the global expression landscape in humans. So this is why we didn't really want to use your approach. Now, on the other hand, if we really wanted to split the data set, that's mean we do not currently have any disease data, but let's say we have disease data, right? That would produce a really confounding result because you would mess a lot of diseases together. Anyway, if we indeed split the data set, we will get a co-expression landscape for each of the biological conditions that we separate. So let's say we use a data set with control and with brains from Alzheimer's patients. In that case, we produce a co-expression landscape that corresponds to the global expression regarding the Alzheimer's disease.
So, can I make one question to clarify this?
Of course, yes.
So, starting from the opposite direction, okay? So Pablo suggested that you split the data. I would suggest that what would happen if you merge the data? If I understand correctly, you said that all tissues are equally represented. So you have some...
That's what we try to do.
Okay. So it is obvious that the correlation within tissue samples, same tissue samples, is much higher than within tissue samples, right?
Sure, but this is why we used a certain normalization algorithm, which does a tissue-aware method of normalization.
Okay, but then when you showed that tree, it was pretty obvious that samples from the same tissue would cluster together as you would expect them to be,
of course,
right? And then you pruned the tree in order to get something that would be more meaningful in say, extracting the correlation, the co-expression relationships in all tissues, okay? So my question would be, why wouldn't someone merge the samples of the same tissue, coming from the same tissue, in one, say, sample that is representative as a mean of this tissue, and use that instead of the whole data set, in order not to have, I don't know, biases coming from high correlation within tissues against within tissues, if you see what I mean.
Yeah. Actually, that is the kind of thing we try to do here, because we try to get, by pruning the tree, we try to get the same number of samples for each tissue. Of course, since that's not realistically possible, perhaps next time we should consider using your approach, as it seems really a nice thing to do. We should try testing that
way. It would be faster, probably.
Especially, that would be especially the case.
And if I make one last technical comment, so you create the UPGMA trees with a distance metric that is 1 minus the Pearson correlation. Did I see correctly?
Yep.
So what happens if you have negative correlation? Do you have negative correlations?
Unfortunately, this approach misses out on that aspect.
Okay, so you don't take them into account?
No, this is only a positive correlation for expression tree.
Okay, but which part of your data is, so what is the percentage of genes in your data set that have negative correlations? How many do you leave out?
Honestly, since that wasn't our main aim, we didn't quite really test that. But on the other hand, we're thinking about new approaches to visualize all those interactions.
Okay.
That would also take account negatively correlated data, things.
Because that would be informative. A strong negative correlation would imply functional relevance, even of the opposite type, I guess.
Of course, don't worry, we are definitely looking at that.
So, there is one more question from, can I see?
Please go ahead.
So there is a question, if you performed any experimental part to prove the results of your tool. So any experimental validation underway or?
No, unfortunately we are a fully computational laboratory, so we cannot perform those ourselves. The only thing we did was verify our findings through already existing bibliography. Although we do encourage anyone who uses our tool to, if he finds an interesting biological hypothesis to an already existing biological question, to try and confirm those data.
Okay. So, thanks again, Vasily. Thank you to all the speakers of this